So when we're looking at this question, it says the tRNA molecule with the anticodon GCU, it says, use the codon chart to be careful. And then to use the codon chart and be careful, period, think, exclamation point. That's what I wrote. So I told you it's an anticodon GCU. What do you think most kids did? The ones that didn't think and weren't careful.
They just thought they was about to like flip it like mRNA.
Yeah, they thought it was mRNA, and they just went and used the codon chart and changed GCU to an amino acid. That's what most kids did. They got this wrong, and that was wrong because I told you it was the anticodon, and the chart, even on the test, it says codon chart. So what did this question test? It tested whether you understood the difference between anticodon and codon. The anticodon is on the tRNA that's carrying the amino acid. The codon is on the mRNA. Both of them are triplets. Both of them are threes, right? So that's what this was testing without me having to actually ask you that. This question. Yes, you may. So the tRNA is GCU. That means that the mRNA was CGA. And that's the one you looked up that gave you a different amino acid than GCU did. So the people that got it right found the mRNA codon and just found the amino acid, and it was pretty straightforward. The next tricky question was if the code for an amino acid is AS or AGC. Yes, jeez. Amino acid is AGC. On the DNA molecule, right? If the DNA molecule says AGC, then it says, what's the anticodon on the tRNA would be what? Well, most kids went and converted again. They just went right to mRNA. They only memorized it in their mind. What came to them is AGC is UCG. That's it, done, boom. But it's actually AGC because this is on the mRNA, and I asked you for what? The tRNA, the anticodon on the tRNA, which is the opposite of the mRNA, which is identical to the DNA.
Oh, right, except on a
Except on a tRNA.
It's a U instead of a T.
Exactly, except on a U, and the tRNA would be U. Exactly, you got it. Give that up, that's right.
Right here.
Don't get jelly. All right. Huh?
The left one up top, what was the last one after CGA?
After here?
My left. You're right.
This, wait, are you talking about this here?
She don't know. She don't even know.
Yes, I do.
All right, so then the next thing that gave people problems were these gene mutations. And remember, I did these frame shift problems with you, and I put them on the board. In fact, there's 2 problems still on the board. I told you, remember, if you insert, I said this, and you can go back and look at the video on YouTube, or you can look at your notes, however you did it. I said, if you insert or delete anything that's not in a factor of 3, right? Not 3 or 6 or 9, right? If you insert or delete 1 or 2 nucleotides, then you're going to shift the reading frame. Remember the sentences I wrote up there? The boy cut whatever. And we inserted 1 letter, and we saw that everything after that shifted. And we deleted 1 letter in the other sentence, and you saw that everything after that shifted, the message shifted. And we said it was a frame shift mutation. That's what happens when you insert or delete anything that's not a multiple of 3, you get a shift frame. Frame shift.
You just kill someone?
Tried to. I don't know if I did.
He does.
So the question that I asked, and the details were different for every test, but the question I asked you was, if I had this gene and it looks something like this, and I had this mutation was shorter, so what kind of mutation happened? A deletion, correct. most people got deletion, they got that part right.
Yeah.
But if you deleted something, what happened? You had a frame shift. So you had to have both deletion and frame shift. That's where most people got it wrong. You put deletion, but you didn't put frame shift. So then mutation, the length was the same as the gene, so you didn't delete or insert anything, right? So what was that? A point mutation. If it's a point mutation, you're just taking a G and a C and you're switching it. You might change the amino acid, but you're not going to. Got him. Give me some hand sanitizer. Thank you.
Don't touch it, don't touch it.
All right. So, thank you. So, hey, I gave it a chance to live. It kept coming for me. It had to go. So, a frame shift mutation, if you switch the amino acid and you're not shifting the whole message, it's not a frame shift. So that middle one only has one answer, not two. The third mutation was longer. So what happened there?
Insertion.
An insertion. So you had an insertion mutation, and if you inserted not a multiple of 3, in this case you inserted 1, what happened to the rest of the message? A frame shift, it shifted. So then it shifted, and it was an insertion, so there was 2 answers there as well.
Period.
See me after class, because if that's true, then I'd have to give you, if you got all 3 and that is true, then I have to give you that point. All right, so please see me after class to make sure I didn't make that mistake.
Wait, what happened?
She said that she noticed that there was a point mutation as well as an insertion. So if that's true, then I need to give her the point. If she put insertion, frame shift, and point mutation, and that's true, then she would have to get the point for that. All right, then the last question, almost everybody got the rest of the questions correct, just for the record. So I was very happy with the results overall. But there was this one.
Oh, yeah.
And this one, I did this on purpose. I found this on a state exam, and I said, you know what, this is a good question. I don't like the drawings, but they need to understand that people are going to draw things differently, and every time you see it, you have to be able to put things into context. And see, that's part of the test-taking skills. You have to be able to look at a problem based on what they're asking you. So if they're asking you about transcription and translation, and they give you these options, and then they give you 4 questions, right? Mm-hmm. What label W, what is label Z, what is label Y, and what's label X? What is each of these? And then they give you 4 questions with 4 different options, and all the options are the same, right? For all 4 questions, there are amino acid, ribosome, tRNA, and mRNA. For all 4 of them.
Yeah.
Then common sense would tell you that those choices are right here in front of you.
Yeah. It's one of
That
each.
one of each. So if this one is mRNA, let's say, then this one can't be mRNA. If this one's mRNA, then this one can't be, right? Mm-hmm. So as you make your choices, your logical decisions, you should be able to get rid of the other options. So even if you weren't clear as to what this is, you should have been able to figure it out. You just using test-taking skills.
Process of elimination.
Process of elimination. I love that. That's correct. So as soon as I see 3 little lines, what am I thinking? mRNA. 3 little line, 3 little line, 3 little line, 3 little. I'm thinking mRNA. I'm thinking codons. Mm-hmm. Here's 3 little lines. Why don't I think mRNA on these 3 little lines? Because it's all by itself, right? Because it's just 3 by itself. That would have to be tRNA. If there's a whole chain of 3s, I'm thinking mRNA. Okay, so this X is mRNA. So now that only leaves me with tRNA, ribosomes, or amino acids.
You know Z is ribosomes.
No.
That's wrong?
That's what a lot of people thought.
I hate
Now wait. It was a good thought, but you didn't follow through what you're thinking. And I know where she's getting this from. I know where she's getting it from. What? She's getting it from the idea that when she looked at her study guide, the ribosomes are always like these little dots on the rough endoplasmic reticulum. That's correct. Right, they are. No, you should think it too. If you just, someone else said it's common sense. Well, see, here's the thing. Common sense is not that common. It's not. That's number one. And number two, it's about logical deduction. It's about context clues. And it's about scale. So think about this. If you have an endoplasmic reticulum, then the ribosomes are little dots. You're right. You're gonna see, it's why the rough endoplasmic reticulum is rough, because it has these little dots on it, because you're looking, big picture, you're looking at the endoplasmic. But here you're looking at just an mRNA strand. If you open up your study guides and you look at the drawings of,
This is inside, so that's the ribosome.
that's right, the mRNA is inside, so this has to be the ribosome, and you'll notice that the ribosomes in your drawings look like this. There's two parts, a big and a small, and they look like that. This has to be the ribosome. Right? So you got it. See, do you see?
That while I was taking my test.
I know, it happens to all of us all the time. It happens to all of us all the time. So this is the ribosome, and again, if we use our logic and our context clues, we have to think. Then the other thing is you have to ask yourself, what does tRNA do? What does tRNA do? Carries what? No. It brings amino acids to the ribosome. And if you look at the drawing, when you get the test, I'll give the test back to you. The drawing actually has two of these next to each other. Actually, has two of these next to each other. I'm gonna zoom in.
You should have put a scale on it.
Well, the test on the state test didn't have it, so I didn't want to add something the state test didn't have.
It looked like.
So it had something like this, and then it showed you this happening. Right? So you had to look carefully. And so here you have these triplet things that you identify as tRNA. You know that tRNA carries amino acids. So these little yellow things, if they were ribosomes, what's a ribosome doing on top of a tRNA? And why are these ribosomes connected together? That's not right. So this is tRNA. We know should be carrying an amino acid, and the ribosome should be linking the amino acids together. So that's what you have here. That's what this is. These are tRNAs. These are amino acids. So Z is an amino acid. This is a tRNA before the amino acid gets linked on. So again, using, yes, logic, but also you have to, what did you have to know to be able to answer this question? You had to think scale. You had to think what's the process? What's actually supposed to be happening? You have to know what is tRNA really doing in this process? What's mRNA doing in the process? If you knew that, then you probably got this one right. Again, it's easy to get things confused. That's the nature of tests. So it's opened up now, and it'll be due tomorrow instead of today, by the end of the day. So tomorrow's Friday. I want to remind you that all the answers to that case study are already posted 3 different times on my YouTube channel. 3 different explanations. You can use any of those. You can just figure it out on your own. Can I do what? Yes, you may. After in turn, take your turn. What?
What? I think we can say what videos correlates with.
Yeah, it correlates with the classic experiments video.
Oh, yeah,
That's what it's called. Just type.
Manuel Mendoza classic experiment.
Yeah, go to YouTube, type Manuel Mendoza classic experiments, and it'll show 3 of them will show up.
It doesn't fuck you famous. Normally I gotta search for everybody else. Go like all the way down to the
Huh? Say what?
Okay, I'll leave.
I don't know. I don't really care. So I only do it for you guys. All right, take your turn. So whenever the next person, you gotta have the bathroom pass. Go get water. The bathroom. There's 2 people went to the bathroom already.
Oh my Lord.
All right.
How do y'all go to this smart school and be so slow?
It's not, they're not slow, don't say that. They're just, people get, we all, hey, I've already shared with you how many different things that I've done that people would call slow. That I myself, looking back at my past, call myself slow. I'm like, how could I be that stupid, is what I say to myself. The reality is that we are all that way. Stop thinking that you're better than anyone else or anybody. We all make mistakes. We all do stupid things. Didn't I share with you what I did in the Army with the gun?
Oh, you almost shot yourself or something?
Oh my God, no. Jesus God, protect me.
No, it was one of them like automatics or something like that.
You told us this. We just don't remember.
All right,
Yeah, like,
so
I remember-
anyways, good
Tell
story,
me the
but we don't have
story.
time. We don't have
Say it
time.
again.
So again, the full explanation on these case studies were posted on YouTube already, but we should try to get some understanding of what we're talking about here. In the 1930s, now, does that sound like a long time ago, 1930? Well, it's almost 100 years ago now. In the 30s and 40s, right? 1920s is 100 years ago. So it's 90 years ago. So in the 30s and 40s, we knew people inherited stuff, right? You heard of this guy named Mendel, right? You did his Punnett squares, correct? You guys told me 100 times you did Punnett squares in middle school. And you didn't hear about the guy named Mendel? All right, so somebody. So Gregor Mendel, he was a monk, an Austrian monk, and he's the one that came up with the P's and big A, little a, recessive and dominant. He came up with that through statistical analysis. So that happened long before the 30s and 40s. So the point is that because of this guy named Mendel, we knew that there was this stuff that was inherited. Does that make sense? That we inherit things. Now that's almost common sense to you, right? I inherited my nose from my mom. I inherited I'm tall because my father's tall. I'm short because my mother's short. Whatever. Aren't we used to that? Come on, focus. So we're used to this idea that people inherit things. But how do people inherit things was really a big question for the longest time. There was an idea in the 1600s that thought of women as a field and that the men planted their seed in the field. And so what that meant was that, understand, and people talk about things like this, about people inheriting things like planting seeds, like farmers all the time, right? And their idea was that there was in the male's gamete, and we call it a sperm, right? In the male's gamete, they thought there was a tiny little human being curled up. There's a little human being curled up, full little tiny baby human being. And when that's what the male was giving to the lady, and then the male decided if it was a male or female, there's either a little guy in there or a little girl in there. And the male was everything was inherited by way of the father, right? So in Europe, obviously this is only in Europe. So this is the concept that was really pushed for a long time, that things were inherited via the male. They were contradictory ideas, but because it really didn't follow along. If you looked at what you saw in nature, that's not what was happening. But that's what people thought. They called this the homunculus. Homunculus. Tiny. Say what? What? Homunculus. Don't ask me to spell it. You know me in spelling, and let alone that word. So this tiny little guy, that's what they thought. Well, after Mendel, we knew we inherited these things. These things didn't get inherited this way. That was not how they got inherited. So after Mendel, we knew that the male, and we'll designate the male like this. The male and then the female that looks like this, they give half of something. And Mendel said it's that they're going to give this thing, it's going to be a big A or it's going to be a little a. Or maybe two big A's or maybe two little A's, right? But one parent's going to give one thing. He didn't know what it was. He didn't know anything about DNA. He was a monk growing peas in a monastery. He had no idea about any of this stuff. But he did a statistical analysis of how, when he crossed peas, how they were inherited. And by doing it that way, he worked backwards. He said things get inherited either dominant or they're recessive. And if you got the dominant, it shows up. And if you have two of the recessives, it shows up. That's all he knew. Today, and especially after this unit that we just finished, you know that those things that you inherit are called what? Genes. And those genes are sequences of DNA inside a chromosome. But the units that you inherit are called what? Chromosomes. So you inherit a chromosome. That's what you get. That chromosome either has the big A on it or it has the little a on it, right? And you get one of those chromosomes from mom and one from dad, and that's how you inherit. We know that today. But we didn't know that in the 30s and 40s. Here's what we knew in the 30s and 40s. We knew that there was a big A, a big T, a big U, and a big, not a C, sorry. Big C and a big G in DNA. We knew DNA had that. We also knew that RNA had A, U, C, and G. We knew there was RNA in a cell. We knew there was DNA. And we knew that proteins had 20 different amino acids. Okay? So we knew that. The question that they were trying to answer is, what is this stuff that Mendel described? This inheritable material. What is it that's passed from mother to child and father to child, right? What is it that we inherited from our ancestors? And so they asked themselves, how could you write this complicated creature that's sitting in front of me? When I look at an oak tree with all the different things that it does, what language would you use if you were making that tree? What language would you use to describe this thing, to control this thing? Would you use a language that had 4 letters, like in DNA or RNA? Or would you use something that has 20 letters? What would you use? I'm asking you. Our visitors, if you're an alien race and you're coming in to design life on a different planet, and you're gonna need the right controls, right? Make this thing tall, make it short, make it red, make it blue. This is what you're gonna inherit from now until whatever. How many letters would you use? Would you use a language with only 4 letters? Or would you use a language with 20 letters? Huh? 20, why 20? I mean, I would too. If you asked me, I would answer the same way. But I want to know why you think you would use 20. That's exactly right. So you have a bigger variety of words, right? Of ways of saying. Like in our language, how many letters in the English alphabet? 26. That lets us write all kinds of words, right? We have a dictionary full of words and each one means a different meaning because we have 26 letters. If we only have 4 letters, it'd be hard to come up with all those words, right? All right, so good. That's exactly right. So scientists actually thought that proteins, a lot of scientists thought protein was the stuff we were inheriting. They thought DNA and RNA was doing something weird. They didn't know what it did, but they knew proteins. They figured protein was the inheritable material. Okay. So they did an experiment. Who's done their homework? Who can describe to me the experiment?
Me?
Yeah.
So basically, I forgot their name, but it's like
Yeah, stahl. That's right. Please listen to this young lady. She's on the right track.
So basically, they use like this smooth thing or like eat it or like they saw what was like, taking on, I guess, from the mouth.
Okay, so I'm really happy with your remembering of your homework. But it was Griffith is the guy that did this particular experiment. Mrs. Stahl did the other one. So, but good that you got the name. Some of your colleagues didn't even have that. So Griffith, and it's right there in the case study as well, did this experiment. Obviously, when I say Griffith, there was a whole lab. It's not a single person. So they were looking at pneumonia. Do you know what pneumonia is? Not ammonia.
Oh, the sickness.
It's pneumonia, the illness, yeah. The stuff, the lung disease. Do you know that it's a bacteria? All right. It can be a virus. Pneumonia can be caused by a virus. But in this case, it was a bacteria. Man, what? Like what? Your lungs get filled with fluid, with water, and because of osmosis. And the bacteria cause this, and your lungs get filled with water, and you suffocate. That's pneumonia. So, but pneumonia can be caused by, pneumonia is the filling up of your lungs with water. It can be caused by pneumoniae bacteria, or it can be caused by a virus. Either one can cause pneumonia. Now, Griffith was studying two types of bacteria. He had one that was smooth. They're the same species, same strain, different strains, but they're related to each other. They're both pneumoniae bacteria. And the other one, though, was rough. It had little proteins, those surface proteins we talked about earlier. Well, it turns out that the smooth one, the smooth bacteria, they produce mucus, like you do when you got the snot, okay? And that mucus protected them against your body's antibodies. So that mucus layer around this smooth bacteria meant that your body didn't even recognize it. Animals like rats and mice, by the way, rats are very similar to us anatomically. That's why we study them. That's why we use them in laboratories. They're very close to us physiologically. And it turns out that the same thing that kills them kills us. And a lot of the same things that kill them kill us. So the smooth bacteria, the rats could not identify it as an invader. And so if they were injected with a smooth bacteria, they would die. The rough bacteria, their immune system could identify, and because they could identify it, their rough bacteria means they survived. Is that clear? You have to understand that or you won't understand the experiment. So now knowing that much, is anybody confused about that right now? Because this experiment will be on the test, right? So you have to understand it. All right. So let's make a chart, and you see that there's a chart there. There's 4 options, right? In the chart. 1, 2, 3, 4. And if you inject with the rough strain, does a mouse live or die? If you inject with a smooth strain, and what happened in the experiment when he killed the bacteria? So if you kill the bacteria, can it kill you? It can't kill you because it can't multiply, it can't invade you, right? It's dead. So they cooked it. When they cooked the smooth bacteria and put it in the rat, what happened? It lives. Now here's the interesting part. The last one's the interesting part. This is a control. This was a control. This was a negative control, right? This was a control. This was a positive control. We knew that this bacteria should have killed the rat, and it did. We knew that if we killed the bacteria and injected it, that bacteria should not have killed the rat, and it didn't. So that's good. And we knew that and this was also a negative control. So these are 3 control groups. Each one, we knew exactly what should have happened. And in fact, that's what happened, right? No, there are no surprises there. If I kill the thing and inject you with it, it shouldn't hurt you because it should be dead already. If I inject you with the disease with the smooth that causes death, then you should die. And if I inject you with the one that shouldn't cause death, you should live, right? Does that make sense? So these three, they're known. We knew, we expected results, and if they didn't, if something went wrong, then this would not have happened. The one that we did not know, the experimental group, was this last one, right? And then what they did is they took the heat killed, so they're killed, and they mixed them with the RUP. Now the RUP does not kill, right? But the heat killed, they were broken up into all their pieces. So DNA, proteins, lipids, carbohydrates, all in that test tube. This bacteria, they're RUP, that are alive, these are alive, don't cause disease, are mixed with the stuff from the disease-causing ones that you've killed. And they sit in the test tube for a minute, or an hour, 2 hours. What happens, what do you know at this point, what can bacteria do? They're really good at multiplying, so these guys will make a lot more of themselves, that's good. What can bacteria do with each other? Yeah. Share the DNA. Share what kind of DNA? Yeah, it begins with a P. It's round, small and round. They are prokaryotes, but they're called plasmids. But, so they can share DNA. We know that today. They didn't know that back then, right? But we know today that they can share stuff. So you and I would expect that the stuff that's in here would move over to the live, and then the live one would get become deadly. That's what we'd expect. But they didn't know what was going to happen. So what happened when they did that? They died. So that told them that there is what? There is inheritable material here. They can't inherit, right? So that's what's important about this experiment. So how would I test this on the test? I will ask you, what are the control groups in this experiment? I will ask you, what's the experimental group? And I will ask you, what did this experiment show? And what it showed is that this bacteria has inheritable material that it can send from one bacteria to the next. Okay, so you have this experiment, and Griffith took, as I just explained to you yesterday, right? Griffith had this bacteria that caused disease. He killed it in a test tube. He heated it up and killed it. Then he injected that into the rat, and the rat lived. So the question is, why did he do that? Like, we know it's a negative control, but why do that? Here's the answer. It's answering this question. How do I know that it's the live bacteria that kills the rat? How do I know that the bacteria doesn't produce a poison that kills the rat? So what would be an example of a poison that kills a living creature? There's something called botulism. Have you ever heard of it? Well, it happens, it's a bacteria called botulism, and it produces a toxin. It grows in food. It can grow in like mayonnaise. So if you go to a picnic, summer picnic, and somebody has mayonnaise, like a potato salad or something, sitting out without being cold, you don't want to eat it, because if the botulism bacteria is on there, it's producing a toxin. And what's scary is that this stuff can grow in a can, let's say, and you open up a fresh can, and you pour it out, and you cook it, right? So you cooked it, you killed the bacteria. But you eat it, it could still kill you, because it's not, the live bacteria is not what kills you. It's the poison that the bacteria left for you.
Alright, so what about, like, you know how, say you have a rat trap and there's a little poison in the food, and
Wait, what?
Rat poison.
Rat poison?
I thought it was just cheese.
Rat poison is a poison it's a hemolytic. It causes the rat to die from internal bleeding. It's a very painful and horrible death. I don't even like to think about it. But look, the point is,
Because you got rats.
well, I do have 2 pet rats, but it's not really the reason. I don't like anything suffering. If I'm gonna kill it, I would rather it die quickly than it suffer a miserable death. It's a good question. Salmonella, the difference between salmonella and botulism, salmonella, if you have salmonella on chicken, which a lot of chicken has salmonella in it. But if you cook the chicken thoroughly, guess what? The salmonella dies and it's not poisonous anymore. They can't grow in you, so it doesn't make you sick. Botulism, if you kill the botulism toxin, if you kill the bacteria that causes botulism, the toxin that causes the problem is still there. So it'll still make you sick. Does that make sense? That's the question. The question is, does this bacteria kill you by making more of itself? Is it required that it be alive to kill you? Does it produce a poison that kills you? Does it cause pneumonia by producing a toxin that causes your lungs to be filled with fluid? Or is it the bacteria itself, living bacteria that multiplies, that causes that? Do you understand the difference? Yes or no? It's okay if you don't. So by killing the bacteria and in heat killing the bacteria and then injecting the dead pieces and parts into the rat, they knew, and it lived. They knew this bacteria had to be alive. There's some kind of living material, not a poison that the thing produces, but it's a living thing that's causing this disease. That's what this new negative control showed. I recorded it, I'll post it, okay? But I'll say it one last time. By killing this bacteria and breaking it up into its pieces and parts, right? When he killed it, they broke it up into proteins, DNA, and RNA, and all the other parts, and they added in a test tube. And then they injected that into the rat, and the rat lived. That showed clearly that the bacteria had to be alive to cause pneumonia. A dead bacteria doesn't cause pneumonia. Does that make sense?
Yep.
So we know that because of this negative control. The next thing they did is they took the live version of this bacteria and they injected it and the rat died. That's the positive control. So now we know the live bacteria kills you. The dead bacteria doesn't. Because it's dead and it's not creating a poison, right? So the last negative control is you took the rough bacteria and injected the living rough bacteria into the rat, and the rat lives. Now you know that the rough bacteria does not kill the rat. So knowing those 3 things are incredibly important for your argument. The rough don't. It has to be the smooth. There's something that the live smooth bacteria has that makes it deadly. The rough bacteria doesn't have that thing, whatever that thing is. We know today it's a mucus layer that prevents the immune system of the rat to attack it. So we know that these 3 here, that gives us really important information so that when we do the actual experimental group, and here's my question I'm going to ask you on the test. What question is the experimental group trying to answer? For this experiment, there's 2 experiments here. This first experiment and then the next one. What is this part of the experiment trying to answer? Yeah.
We just passed on DNA, RNA, or amino acids.
Good. That's the big picture, right? But in this particular part, this doesn't answer that, right? This part is just answering this question. Are you ready? The question they're trying to answer here is this stuff that is deadly, the stuff that kills the rat, can it be inherited? Is it inherited?
That's why it's an experimental.
That's what the experimental group's going to answer. So how do we figure that out? Well, we take the heat-killed bacteria, so it's killed. And what do we know about bacteria? What can they do that we can't? What can prokaryotic cells do that eukaryotic cells normally don't?
Asexual.
transfer genes asexually through something called plasmids, right? So we knew that they could do that. We knew that they had this ability. Well, we know today that they can't. But we had a question, is there something in the words they used, Griffith used was transform, right? This transforming molecule, this inheritable molecule. Is that something, can you inherit deadliness? Can you inherit the ability to cause pneumonia? That's the question this part of the experiment's answering. So he killed it, and then they allowed it to sit in a test tube with the rough. We know the rough doesn't kill the rat. We know that the heat killed by itself doesn't kill the rat, right? Correct? So we leave them in the test tube by themselves for a while, then we inject that in the rat. What happened to the rat? They died as if these things were smooth. And when you looked at the bacteria under a microscope, they were smooth. They were rough until they were exposed to the stuff that's in the heat kill. So what did that tell Griffith? What did that prove? Not prove, but what did that show Griffith? That something transferred. Today we know it's a gene, correct? But then they said something got transferred. Something got transferred and it transformed. It changed the bacteria. It changed what this bacteria was, from rough to what? To smooth. Again, today we know that was a gene that got transferred, but he didn't know what it was then. But see, this part of the experiment was very important. If he wouldn't have done the control group, then he could not make that argument, correct? Because he could have said, well, how do you know some poison didn't kill the bacteria that was left over from killing the smooth? How do you know that the rough bacteria doesn't kill the rat? How do you know that the smooth bacteria kills the rat at all? That's what these three control groups did. And when he mixed these together and it killed the rat, he and his colleagues knew that there was something that this smooth bacteria had that could be transferred, that could be inherited, that would change the rough bacteria to a smooth bacteria. Today we know it as a gene, but back then they didn't know what it was. Are we good? Yeah. That's what. So if you understand that, you're gonna be able to answer the questions on the test next week easily. We had another tool in our arsenal back then. We had these enzymes. We had these things called proteases. We had these things called DNA ACE, right? Oops. DNases. And we had these things called RNases. What do you think these things did? Proteases did what? Speed up the process is good because they're enzymes. I love your thinking. That's perfect. They speed up the process. What process do you think proteases sped up? Breaking down a protein. It could be the breaking down, but if you read the experiment, which obviously you haven't, the proteases increase the breakdown of proteins. But you know what? I want to point something out. You didn't do the homework, so you didn't know this, but what's really cool is you used your mind, right? And you said it must speed up the making of proteins. Because it could either speed up the making or the breaking. You made a guess and you were wrong, but it's cool that you thought that you got that close. So these things break down proteins. They break them down. In fact, we add proteases. You can go to the grocery store and they call them meat tenderizers. And you can sprinkle it on your meat. That's what they are. They break down the protein. So the meat is protein mostly. It breaks down the protein and makes it easier to eat. Maybe, yeah. Yeah. What are DNases? Yeah. Break down DNA. Is this hard? It's really kind of straightforward. And what do you think RNases do? Thank you. So that's very useful. So And you'll see, I think you'll see why this experiment was so beautiful. Because they knew that the bacteria could inherit something, but they didn't know if it was a protein, DNA, or RNA. They were guessing it had to be protein because we had 20 letters in protein, so it has to be a protein, but they didn't know. So you can imagine how they use these tools to design an experiment, but I'm gonna go ahead and tell you because we're done. We kind of got set back a day. So the first part of the experiment, they took the test tube with the live bacteria and then the rough bacteria. This is the rough, right? And to the rough, instead of adding just simply the dead bacteria, they added the dead bacteria they had DNA, proteins, and RNA, and they added RNase. What did that do? What did this do? Broke down all the, so there's no RNA in here, right? Right, there's no RNA. So if this rough becomes deadly, then we know what? Is not the inheritable material. Because if this rough strain becomes deadly, if the rat dies, right? If it dies, then this bacteria inherited something deadly, right? But it couldn't have been RNA because we got rid of the RNA. Am I making sense? So this thing did die. So they knew at that point it was not RNA. So that's right, could. So the next thing they tried was proteins. This is the one that we're all excited about. So they added the dead bacteria to the proteases. Then they added that to the rough strain. Again, this one they expected for sure. They said, this rat's gonna live because it can't be, it's the protein, they thought. It has to be the stuff that they inherit. But what happened to that rat when they did that? It died. They were very surprised. They were very surprised. They said, oh my God, it's not proteins. The stuff that's inherited is not protein. So the next thing they did, what do you think they did next? The DNAs. So they took and they got that dead bacteria and they added DNAs to the rough. And now what happened? It lived. They knew because they got rid of it and it lived, but it died with the protein and the RNase, the protease and the RNase. They knew that it had to be DNA that was inherited. This was so do you see the elegance of the experiment? How clear the results. You can't argue it, right? It might be something else, but we know for sure it's not protein, it's not RNA, and it's probably DNA. It is common sense. But see, common sense is not that common. All right? And common sense is not that common, and science is nothing but common sense applied in a methodical way. So it's just really a beautiful experiment. So that's how we know that DNA is what's inherited. And of course, you've already learned about genes and promoter regions and et cetera. Tomorrow we'll go over how DNA is replicated. How do we make copies of DNA? And then Monday we'll start with inheritance of chromosomes and manipulation of genes. Very good day today. Thank you. So the second experiment had to do with this guy, meselson. You can have this back. I just need to write this down. meselson, but just take notes now, and stahl. These are professors, and they did this research, and they looked at what about DNA and how does it work? There's this guy named Chargaff. It's Chargaff. And Erwin Chargaff was an Austrian-Hungarian guy. He's in Europe, right? Eastern Europe. Northeastern Europe, and he was a biochemist. And what he came up, because of his work, it took him years of hard work to figure this out, but you've memorized it quickly, and you've used it on your exams and your quizzes. And he came up with these rules, and you've used them for the last 2 weeks. He came up with ATGC and AU. All right. The base pairing rules he came up with. He came up with a rule that said, it turns out if you have 20%, if DNA has 20%, and please pay attention because I'm going to ask you a question like this on a test in the near future. If I have 20% A in a DNA sample, what percentage of the others do I have? Go. If I have a DNA sample and 20% of the DNA sample's A, what's the rest? What is it? No. What is it? Wait. What is it? 20% A, then what? I can't hear you.
20% G.
Why 20% all of them?
That will only be 80.
I need you to look at this board and think about it. It has to equal what? Yeah, but it has to equal what? What percentage of, if I have 20% A, what must something else be?
40%.
You got it. Somebody else figured it out. You had to put some thought into that. 20% A means what? 20% what? T. No. You're not thinking it through. Listen. 20% A means you have 20% T. That's correct, because for every A you have a what? A T. And I got 60 left. So what are the rest? 30 and 30. So this simple rule that you have learned, there are implications. It's not just learning that A and T, A goes with T and G goes with C and A goes with U. Yeah, okay. And so, right? That goes back to the test you just took. A lot of you memorized. Insertion means this. Deletion means that. But you never thought about what does that mean. Even though I told you it shifts the reading frame, you stop there. You have to ask yourself all the time, what does this information mean? So if I know I got A, for every A I have a T, and I know I have 20% A, then I have to have 20% T. Well, that leaves me 60%. So G and C must be 30% each. All that I can figure out with just that simple rule that you all knew already. So meselson and stahl, they had that information. They knew Chargaff's rule, but they didn't understand how you can make copies of DNA. They knew that if you had this DNA, you have 2 strands, and we all know what that looks like now, right? He has 2 strands. They're all double helix. They didn't know it was double helix at the time, but they knew that they go together. They know for every A, they had a T on this side. For every G, they had a C on this side, correct? They even knew that there was 3 hydrogen bonds here between these 2, and there was 2 hydrogen bonds between A and T. They knew that as well. So they had that information. But what they want to know is, how do you make copies of this? Can you think of 3 different ways? Because they came up with 3 different ways. If you did your homework, if you watched the videos, if you paid attention to all that, then you know what the 3 ways are that are possible to make copies of this. Think about it. If I have, let me just make this. Let me just say that. Why don't we do this? Why don't we just say C, G, and let's make, that'll be our code, all right? AGC. How do I make so this one here is AGC. This one here is TCG. Do you see that? How is this copied? How are these 2 long, 3-meter long molecules of DNA, billions of nucleotides? How do I make copies of this? Give me 3 ways I can make copies of it. Yeah. All right, so semi-conservative. And I'm glad you guys did some homework. That's good.
Conservative and
What does that mean, semi-conservative?
like two, it's like 1 did break 2, right?
Yeah. Oh, is it time to go? Yeah, it's important you get out here on time. All right, well, continue watching. The test will be on Tuesday, so we have Monday to go over this. Have a good one. So the stuff from the case study, semi-conservative, what's the other one? Conservative. And dispersive. They sound like fancy words, don't they? But honestly, it's just common sense, really. Bless you. If this is a long copy, right? Now think about what I'm saying. If this is a long copy of AGC, right? It keeps going, right? All the way down this little thing. Bless you. And then the other one also keeps going, correct? So you got this long chain, you're gonna make a copy of it. Think about it. How would you make a copy of it? What are the different ways you can make a copy of it? So I'm gonna make 2 new strands, right? If I have this and I'm gonna make a copy of it, I'm gonna make 2 new strands. And they're gonna be copies, so they'll be exact copies of one another, theoretically, right? So they're, well, yeah, except for mutations, which we've already learned about. Correct? So that's what we're gonna do. But how do you do that? Do you take and take this one strand and copy it and stick it together with another strand? Or do you take parts of this? Like, you can imagine, I think, if you use your imagination and you think about it, I think everyone in this room can imagine that there'd be a bunch of different ways in which you could do this. You can take A, G, and C, and then you can replicate another piece, and you can add a new piece of DNA, and then you can add the old piece of DNA, just kind of ligate it together in different stages, and then do the same thing on the opposite side. So you can do the T, the C, and the G, and you can then add some new DNA here, and then leave some of the old DNA there. That's what they call dispersive. In other words, you can make copies, add new DNA to the old, and copy the old one and make so that you get a mixture of old and new DNA in each strand. You can do that. That's one way of doing it. We don't use this. Nothing that we know of does it this way. But this is just theoretically. Because remember, they didn't know, right? They said, how could this get done? And they said, this is one way it could get done. Then they said, there's another way. I could take the original, the A, G, and C, and I could leave it completely the same. The AGC and the TCG could stay the same, and then I would just make a new molecule of DNA that's an exact copy, ATGC.
What's that called?
And that would be called conservative, right? That's conservative. In other words, you have both the original DNA's molecules stay together, and your new DNA is exact copy, but they're both brand new. Dispersive is when you have part of the old and part of the new DNA strand all in there.
Wait, explain conservative again.
Where you have the original DNA, right? But your original DNA, they stay together, the DNA strand, right, stays together. And see the two originals? They're still together, correct?
Yeah.
Where here, the two new ones are together. So that's why they call it conservative. In other words, you conserve the two strands together. Dispersive means you're making copies. The old and new are coming together. So, by the way, I didn't finish this. I should have finished. So what you'd have is you'd have like something like this. We'd have new and old. Right? And you'd have T, C, G. And of course, the other would do the same thing, correct? You'd have new and old. Sorry. So then again, you'd have A, G, C. So you see, you have your cop. Ooh, I hate when I do that. I apologize. So you have your copies where you see that this is a combination of old and new DNA, where these DNA strands stick together and these two, the new ones, are together. And then you have semi-conservative, where you can take an old strand and you can make a new one. And then you take one of the old strands over here and you make a new one. So you got one old strand and one new strand. One old strand. Oh. And one old strand and one new one. So this would be what? Semi-conservative. So you could have conservative, semi-conservative, or dispersive. And their question was, which one of these three do cells use to make copies? Semi-conservative is the answer. For sure, it's semi-conservative. Don't get confused. But how do they know that? Well, they did one of those classic experiments. Could I have the copy of yours? Because all my copies were taken. So they did some really wonderful work, and it's really just, again, a beautiful experiment. It's going to take a little bit of effort, though, to understand it. Again, I explained all this in the videos that were already posted, 3 different videos. If you get confused or you need further details, you can also find the explanation somewhere, different places on the internet. What is DNA made of, guys? Nucleotides. What are nucleotides? What's in a nucleotide? What elements are in DNA? Say it again. Nitrogen, phosphorus, carbon, not carbon, but carbon, right? Oxygen, correct? And hydrogen, right? Is there sulfur in DNA? It's almost SNOPs, but not, so it's not sulfur. Well, they knew that another word for the nucleotides is nitrogenous base, so they knew that there was nitrogen in there. So that was important, because there's this thing called a radioactive isotope. I think you learned about it in Mr. Custer's class. Yeah. And normally, nitrogen, it has 14 neutrons. All right, so you normally have, 7 neutrons and 7 protons. You have nitrogen 14. That's most of the nitrogen in the universe. It's very stable. It's not radioactive. But there is a heavier nitrogen. It's called nitrogen 15.
And it got one plus, it got another.
It gets an extra neutron. All right, so nitrogen 15 is something that's heavy. And because it's heavy, we could use it. And here's how we're going to use it. First of all, it's going to be we're going to make sure we dye we have a dye. We got these two things. And we also have something else. It's a dye that binds to DNA. That means it's going to change the color of the DNA. So wherever the DNA is in the test tube, we're going to see it. Today we can make these things glow in the dark, but back then,
We should do that.
it costs money. Is unfortunately we just don't have. I wish I could, but I don't have the money for it. I don't know, but I know I don't have it. So what they did is they took bacteria. And they grew the bacteria using nitrogen 14. They had, the food source had nitrogen 14. That's normal, non-radioactive. And so what happened? The bacteria grew. Then they took it and they isolated it. And what they found was, when they added the dye, that there was a band. They saw a band at a specific height from the bottom of the test tube. They centrifuged it and they saw it. It was sitting there. This was the dye color. Let's make it yellow so you know what it is. So now here you know that's DNA. That's what, with nitrogen 14. Then they did it again. They did the same process again, but this time they had what? Nitrogen 15. And what they saw, what do you think they saw? Well, in this case, just looking at it, what would you think would happen if you just had nitrogen 15?
The band moved.
Moved up or down?
Up.
Why down? It's heavy. Right. So that's right. It's okay. That's good. It's good. This is good. We're working. So it's gonna be further down because it's heavier, right? So nitrogen 15 was down. All right. It does. It made sense. That's why it's such a beautiful experiment, because it makes sense. That's when experiments make sense and the results are clear. That's what makes them beautiful. It makes them exciting. The simplest experiments are the most exciting ones because it's very hard for anything else to get confused. So now we want to know how DNA is copied, right? So what we're going to do, what they did, is they grew the bacteria nitrogen 14. So they had normal bacteria growing in normal nitrogen 14. So he grew the bacteria in nitrogen 14. All right. So when they took a sample out and they looked at where the DNA is, they would find it here, correct? At that stage, at stage A. And let's say this is B. Are we good? All right. So now they let this bacteria that all the DNA was nitrogen 14. They let it go through one round of DNA replication. They let the DNA copy itself one time. And after you let it copy one time, they took the DNA out. Where do you think you found it? Well, there was three possibilities. Let's say it was dispersive. Where would you find a band? Because now I want to make sure you have to understand. I want to make sure I think I wasn't clear. So let me be clear. They took a sample out of the stuff they grew in nitrogen 14, and they put it in nitrogen 15, and they let it do its DNA replication one time. So you had a DNA molecule look like this. All the nitrogen was nitrogen 14. Then you let it copy itself. Right? But the new DNA is going to be what? Nitrogen 15. So it's going to be heavier. So now you take the sample. What would if it was conservative, what would you think, where would you find the band? That's right, you'd find it at 14 and at 15, you'd have two bands, right? Because if it was conservative, then you'd keep the old nitrogen 14 bands would stay there together, and the nitrogen 15 would be together. So you get two bands, one band at A and one band at B, right? One for the nitrogen 15. If it was conservative.
And nitrogen 15 would be at the bottom.
That's right. Nitrogen 15 would be down here. The new bands would be down here. The old ones would be up here. All right. But what if it was dispersive? They would, assuming the process, you'd expect there'd be only one band, probably somewhere in between the two, right? Somewhere around here somewhere. Not at A, not at B, but in the middle somewhere, right? Because it's all mixed up, who knows? Maybe you get more bands, maybe one, but something would happen like that. And if it was semi-conservative, if it was semi-conservative, what do you think they'd expect? Somewhere in the middle? Okay, anybody else? So semi-conservative, you'd also expect in the middle, right? Because that's right, because remember, what are we talking about? One old and one new. So it's not going to be light or heavy. It's going to be somewhere in the middle, right? And there's only going to be one band. It's going to be in the middle. It's not going to be A or B. It's going to be hard to tell the difference between dispersive and semi-conservative, isn't it? Yeah, good. I'm glad you were about to say that, because that's right, it would be. But if it was dispersive and we did it again, if we did it one more time, again with nitrogen 15, right? And we did it again, let them go through DNA replication one more time, we would still expect to see somewhere one band, right? Somewhere in the middle, right? Not light, not heavy. Or maybe it'd be a little heavier, but it would all be dispersed, correct?
Yeah.
What would you expect here in the semi-conservative? What would happen? What do you think would happen?
Is it going to be two bands?
Yes. We'd expect two bands, but why? Let's look at why. And this is why it's so beautiful. It would be two bands, and it's the why that's really pretty cool.
Because the O will go back with the O.
That's right. That's right. So you got this old strand, right? And you make new DNA, and the new DNA is going to be yellow. So you get a semi-conservative. You have an old and a new, and you have an old and a new. The old one's light, the new one's heavy. You let this do replication again. What's going to happen?
The O will go back to the O and the new one.
Well, not back. Think about it. You're still growing in nitrogen. You're still growing in nitrogen 15. So then you still got an old strand. It's the new one's still going to be heavy, right? But what about this yellow one? It's going to be with what? The heavy nitrogen 15 is going to be with another heavy nitrogen 15, right? Does that make sense? Because this one was heavy. When each of these is going to go with a new one now. The old one's still nitrogen 14, but the new one's going to be nitrogen 15. The new one, the one that was made of nitrogen 15 from the second round of replication is going to form two nitrogen 15 ones. A new one with both made of nitrogen 15. So this is going to be a heavy band down low. This will be in the middle still. And of course you get two of them, right? Because
So one won't be at A, but
this is semi-conserved. This is semi-conserved. So half of it is conserved. So what there'll be when you look at the bands, what you would expect to see is two bands, one at nitrogen somewhere in between A and B, right?
and then the other one going to be at B.
And the other one's going to be at B, is correct. Because it's heavier. Because this is gonna be the nitrogen 15 version. This is the one that's gonna be 1/2 nitrogen 15 and 1/2 nitrogen 14. So what this proved is that every time you copy DNA, you keep one molecule the same and you make a new one that matches it. That's what this showed. All this work, all these many years of hard work just to figure out that it was semi-conservative, let alone all the other details that we've figured out since then. This process is called semi-conservative. What that means is that you can have one old strand with one new one. The others were conservative or dispersive. Conservative meant that you kept the old and the new, the old ones stayed together and the new ones stayed together. Do you see that?
Right, so the two would add A and B into this person.
That's right. So you see, this is all gonna be new here. They're both new ones here. And so if it was conservative, what would have happened was that you would have had one band at the original A and the other one at B, but that never happened. All right, again, all this is explained in detail in those videos that were already posted, and I will post this tonight. All this will be on a test on Monday? Or Tuesday. I don't remember the day I said, so Tuesday. Very good.
I think I remember the Wednesday.
What? See, that's the kind of game that makes me not believe anything. So Tuesday it is.